United States Department of Agriculture.
Oh, well, thank you for including me in this. I've actually learned so much at this symposium that I'm going to hopefully apply to our own research here in the Cancer Center. I think I don't need to remind this audience that we all follow the guide for the care and use of laboratory animals at our institutes very carefully. But one of the aspects of the guide that is generally underappreciated in my experience is that mice and other small rodents are housed at a relatively cool temperature. People who take care of mice are very aware of this because if it were any warmer, they would be way too hot as they spend hours taking care of mice in the laboratory. And there's other good reasons for why the temperature is lower than what is called the thermoneutral temperature for mice in particular. So in general, and in my experience, most laboratories around the world, and this includes laboratories in very warm climates, 12 months a year, as well as laboratories far north, such as Finland. Generally, the temperature is between 21 and 23, with an average of 22 degrees centigrade. And this is how most cages of mice appear during the day when we are doing most of our experiments. These are Roswell Park bedding, where there's a little bit of enrichment material in the bedding. And the mice generally begin to unwind that material and try to assemble nests to keep them warm. But basically, they use a behavioral response of huddling to keep their environment as warm as they would like it to be. And the recommendation in the guide actually comes with an asterisk that is generally overlooked. And that is that animals should be provided with resources for thermoregulation to avoid cold stress. But this is, in general, not done to a sufficient degree. As I showed you, the typical bedding has a bit of material for nesting, but by the time they assemble this into a nest that they like, it's time to change the cage again. So without nesting material or other forms of enrichment, mice cannot behaviorally thermoregulate. They can't choose or adjust their ambient temperature. They can't build an adequate nest with the typical amount of bedding that's provided. But we're going to hear some fascinating and elegant studies by Brianna Gaskill related to this aspect in the following talk. And in general, they cannot engage in sufficient physical activity, which would also help them to metabolically increase their ambient as well as their internal temperature. The consequence for this is something known as adaptive thermogenesis, which mice turn to almost immediately. And this is a big difference between mice and humans, which humans do use adaptive thermogenesis, but it takes time at a cooler temperature. Mice have the ability to metabolically shift almost immediately. So to remind you what the adaptive thermogenesis is, it's the amount of heat that needs to be produced if an animal is not at the ambient temperature in which their metabolic rate is very low. So cooler temperatures require metabolic heat production if there's not an ability to behaviorally regulate, for example, as with bedding that I mentioned earlier. So at the standard temperature of 22 degrees, mice turn on, they upregulate their basal metabolic rate very quickly, and their metabolic rate increases. That increase in metabolism is lowered if the ambient temperature is brought to near 29, 30, 31 degrees. This varies with different situations. But the important point about this slide is that mice increase their energy expenditure 2 to 3 times their basal metabolism to generate enough heat to maintain a normal and constant core body temperature. How does that happen? It's through norepinephrine in general and the sympathetic nerves. Becoming too cold is a threat. It's a fight or flight response. And the sympathetic autonomic nervous system, particularly the sympathetic nerves, are activated and they produce norepinephrine. There's a lot of beta adrenergic receptors on brown fat, but heat production also occurs in other organs. And what happens when norepinephrine binds this receptor is that in the mitochondria, the uncoupling protein is increased. And what happens then is that during oxidative phosphorylation, protons pass through. When you normally would get ATP, you don't, and instead you get more heat production. And so this is the basis for why sympathetic nerves are activated in our control animals by housing them at 20 to 22 degrees. And our work has focused on what the consequences of this change is in various models. And in our case, it's particularly what happens when we use mice to model tumors. And this is an example of what I'm talking about. This is simple infrared camera analysis of a mouse housed at standard temperature. We call it ST, or 22 degrees. And you can visually see the heat production in the brown fat in the back of the mice that decreases and disappears if you simply house the mice in a warmer environment, in this case, 30 degrees. Some time ago, we tested mice housed at each temperature and found that their core body temperature was essentially the same. This was surprising to me. I didn't know enough mouse physiology to realize what was going on. But now we know that at standard temperature, core temperature is maintained because of adaptive thermogenesis, and that is reflected by a substantial increase in the amount of circulating norepinephrine. Again, in normal control animals, this difference exists. Besides our own work on cancer, we have done several literature reviews to highlight a variety of papers that have come out over the past 10 to 12 years since we published our first cancer work, showing how many different disease models appear to be influenced by the temperature at which we house mice. One of the things I want to emphasize here is that we're not saying that data obtained at 22 degrees is wrong. It's just different. It's a variable dependent upon temperature. And that at 30 degrees is probably not what we want to do to mice either, because in general, being heat stressed is also a very big problem. And we can talk about that at the end of my talk. And I'd love to hear some ideas about how to proceed with the differences and the bias that we are obtaining in our data based on housing conditions. This is the basis of our cancer interest, is that essentially there is a difference between tumor growth rates. If you house mice at thermoneutrality, generally the tumors grow slower. These are two different models in BALB-C mice and two different tumor models in C57 black 6 mice. And all four models demonstrate the similar reduction in the growth rate of tumors. And fascinatingly for us, this difference, this 4T1 model again, completely depends upon the existence of an adaptive immune response. So if you repeat these temperature difference experiments in SCID mice, the tumor growth rates are essentially the same. And when you look at tumors that are grown in warmer temperatures, what we find is a significant increase in adaptive immune cells. So it makes sense that adaptive immunity appears to be important in the control of tumor growth. It's just better if the animals are warmer, and this is quantified here where you can see a significant increase in the number of CD8 positive, the tumor killing T cells in the field of mice housed at thermoneutrality. There's not only more T cells, they're better. They have a more activated phenotype in terms of measuring activation markers such as CD69 or the amount of gamma interferon that these cells are producing. And excitingly, early on, we got this clue that metabolism might really be the difference in these animals because animals housed at thermoneutrality had a lot more GLUT1 on their surface. And so this suggested a better ability to bring glucose into the cells and stimulating glycolysis or oxidative phosphorylation. And so one of our overall hypotheses is being too cold sends a signal to reduce energy usage in systems of a lower priority than thermoregulation. And apparently the anti-tumor immune response is not as important as staying warm enough in these animals. Metastatic potential is also reduced when animals are cooler versus there's the standard temperature and the thermoneutral temperature. It's not prevented, but it's significantly reduced in terms of the rate of metastasis in this model. So because we're dealing with sympathetic nerves and norepinephrine and beta adrenergic receptors, we were able to fully take advantage of the decades of research on beta blockers, such as propranolol, or beta agonists, such as isoproterenol, to mimic the conditions of temperature. So at thermoneutral temperature, we can use a drug like norepinephrine. And at standard temperature, we can block adrenergic signaling with propranolol to mimic the effects of thermoneutrality. And we also have a variety of beta adrenergic receptor knockouts in the lab. And so this is an example of the use of propranolol, a beta blocker. So again, this whole experiment was done at standard temperature. And you see a difference in tumor growth rate that looks very similar to standard temperature versus thermoneutral, but it's not. Both of these animal groups are maintained at standard temperature, the cooler temperature, but propranolol is blocking the ability of norepinephrine to bind to brown fat and produces heat production. And again, this phenotype is completely dependent upon the adaptive immune response. It's not just transplantable tumors, but here is an example of a tumor cell type for ovarian cancer that's growing in the intraperitoneal cavity. And even these aggressive single cell cancers are reduced in the presence of a thermoneutral temperature. And again, if we treat with propranolol in this model, again, peritoneal ascites tumor growth, we still see the effect of propranolol in terms of reducing tumor growth. I want to just quickly summarize now some of the immune studies that we've published. We are very interested in the role of adaptive immune response cells such as CD8 T cells, which we already know that norepinephrine block. But we are also interested in the other arm of the immune response, and that's the immunosuppressive cells, such as M2 macrophages or myeloid-derived suppressor cells. So we've looked at each of these major arms in terms of the impact of cold stress. As I already alluded to, that in cooler animals, there is apparently a suppression of metabolism that makes these cells resemble what we call exhausted immune cells in the tumor microenvironment. And blocking that stress increases T cell receptor signaling, glycolysis, oxidative phosphorylation, and the presence of CD28, for example, a co-stimulatory molecule. In the case of suppression, this is a model where we're using this in the beta adrenergic receptor 2, beta 2 adrenergic receptor knockout animals. This is an interesting way to do these experiments. There's, again, no difference in room temperature here, but simply the absence of beta adrenergic receptors gives you a similar phenotype. But now in animals that have no receptor for norepinephrine binding, so it's similar to putting the animals in a warm temperature where norepinephrine goes down. Now we find a reduction in the number of suppressive cells. So there's a balance going on here that's very bad for tumor growth. We have poor activation of CD8 T cells, but we have a greater frequency of immune suppression going on. And I won't go through the details of some of the papers where we've characterized the mechanism by which norepinephrine binding enhances the survival and immunosuppression potential for these myeloid-derived suppressor cells. So this may be the mechanism by which the system says, I need to stay warm. There's an energy draining process going on here, tumor growth, and I need to suppress the amount of energy that's going to the immune response. The frequency and function of MDSCs may be how that happens. So to summarize, we have what we believe is a useful model system for looking at the impact of thermal stress, but it's also a system, an example of how two different experimental results can be obtained based on environmental conditions. Neither is wrong, but doing one without the other, we believe, biases our impressions of tumor growth rates or the efficacy, for example, of chemotherapy, radiation, and immunotherapy, which I haven't had time to present today, but we've published on each of these. And in fact, with regard to immunotherapy, we've taken some of these ideas to clinical trials. And as I mentioned at the beginning, being cold is one of the fight or flight responses that are similar to chronic psychological forms of stress, such as anxiety, fear, and depression, that are undoubtedly increased in our patients with a cancer diagnosis. So while we're not evaluating whether they feel cold or not, we're simply assuming that stress in the autonomic nervous system is increased through a variety of chronic stressful experiences that are mediated through the sympathetic nervous system. So we have initiated and started to complete some clinical trials where we're adding propranolol to immunotherapy, such as checkpoint inhibitors, and also to radiation and chemotherapy. So what to do? And this is where I have learned a lot at this meeting, and I'm hoping for ideas from others in the audience. What can we do about the recognition that housing temperature may be a problem? I don't think that 100% time at thermoneutrality is a solution, because thermal stress can occur. And in fact, that's the main reason why the guide suggests a cooler temperature. But what the guide really didn't predict was that if you're modeling diseases in mice, there may be an even greater cold stress going on. I think mouse bedding modification is a good solution. If you have the ability to have enough nesting material, you allow a choice. The animal can become warmer or can move to a part of the cage without as much bedding. And this is work that's been pioneered by Brianna Gaskill that we'll be hearing from. Housing density modification, such as what Karen Svensson and Beverly Pagan have been studying, I think would benefit thermal stress if we were allowed to put more mice in a cage. Their huddling may be more sufficient for staying warm enough. And we may be able to modify a corner of the cage. We've published some preliminary work where we were able to heat just one corner of the cage. And I think part of this will require some different types of caging systems. that allow choice so that when an animal feels cold, it can move to a warmer environment. But what can be done right now, I do think that we need to conduct selected experiments at the two different temperatures as a control for data. And we may be able to conduct small pilot studies at different temperatures and in the manner that we've just heard about. And I think in the meantime, we must start reporting better the cage temperature, the room temperature, and the humidity of the experiments that we're conducting. And I'd like to just end with thanking many graduate students in my lab over the years who have been doing this work. And I'd also like to highlight my clinical collaborators who have taken this work to the clinic, where we're beginning to learn from our animal models that chronic stress is a bad thing in cancer. So with that, I'm going to stop and thank you all for your attention.
USDA is an equal opportunity provider, employer, and lender.